Good evening to everyone. I am Alessandro Pellizzaro. I'm a biologist and I have been working in Acque Chiampo S.p.A laboratory since 2004. The study is part of my PhD research born from a collaboration between the University of Padova and Acque del Chiampo S.p.A. Acque del Chiampo is the in-house manager of the integrated water service for 10 municipalities in the province of Vicenza. The aim of the study is the evaluation of perfluoroalkyl acids uptake by hydroponic crops and emerged macrophytes. Are usually classified in perfluorocarboxylic acids and perfluorosulfonic acids depending on the functional group carboxylic or sulfonic. We studied 14 PFAS, 11 perfluorocarboxylic acids with 3 to 13 fluorinated carbons and 3 perfluorosulfonic acids with 4 to 8 fluorinated carbons. The objectives of this study were the evaluation of PFAS model uptake by different hydroponic crops and the assessment of PFAS uptake by aquatic macrophytes in order to estimate the human exposure through the food chain and in order to assess wastewater phytoremediation by selected plants. Two experiments were performed in an unheated greenhouse located in the Arzignano wastewater treatment plant area, hydroponics by horticultural crops, and constructive wetlands mesocosm by emerged aquatic macrophytes. Plants were exposed to fast-spiked solutions and wastewaters. All waters were added of nutrients and at the end of the exposure period, we determined PFAS concentration in roots and leaves and bioaccumulation factors values. In order to determine PFAS concentration in vegetal biomasses, at the end of the treatment period, plants were divided in shoots and roots and dried in the oven. Dried biomass samples were wicked and treated with methanol. The extracts were concentrated and passed through cleanup cartridges before the analysis in a triple-quadruple mass spectrometer. Of PFAS concentration in roots and leaves and the vegetation dried biomass, plant uptake and translocation of PFAS were determined according to different bioconcentration parameters for all perfluorinated compounds. Root concentration factor and leaf concentration factors were respectively calculated as the concentration of a compound in roots and concentration in leaves divided by its concentration in solution. Translocation factor was determined as the concentration in leaves divided by concentration in roots. Hydroponic test was performed by deep flow technique in a recirculating hydroponic system that hosted 60 plants of lettuce and spinach. 12 plants per batch and per species were analyzed after the exposure period. In a batch, lettuce and spinach were fertigated by the same solution. Hydroponic test was performed by 2 sessions. 2 synthetic PFAS mix solutions, hereafter called Lab 500 and Lab 5000, and 3 wastewater treatment plant effluents were tested. And in this table are reported all PFAS concentrations. This bar graph shows PFAS concentrations determined in roots and leaves of crops treated by Lab 500 solution. We can observe that most of PFAS concentration in roots were higher than leaves for both crops. Most of PFAS concentration in lettuce biomasses were higher than spinach. Kilograms represent root concentration factor in logarithmic values of each compound in both crops treated by Lab 500 solution. We can appreciate the sigmoidal pattern of perfluorocarboxylic root concentration factor and the increasing of perfluorosulfonic acid root concentration factor with the increasing of perfluorinated chain length. Graph shows translocation factor from roots to leaves of both crops treated by Lab 500 solution. We can observe that translocation factor decreased with the increasing of perfluorinated chain length. Furthermore, translocation factor of spinach resulted always higher than lettuce. Graphs report perfluorocarboxylic acid root concentration factor values in all water tested in hydroponic experiment. We can observe that root concentration factor of perfluorocarboxylic acid in wastewater treatment plant effluents showed the same pattern of lab solutions, and the perfluorocarboxylic acid root concentration factor values were similar among the test sessions. This is very interesting because similar root concentration factor data were determined by different water treatments, different crops batches, and different climatic conditions. We can observe the same pattern of similar values between root concentration factor of perfluorosulfonic acids in all waters among the test sessions. Summarizing the hydroponic experiment, root concentration factor of perfluorocarboxylic acids showed a sigmoidal relationship with the perfluorinated carbon chain length. Perfluorosulfonic acids root concentration factor increased with increasing of perfluorinated chain length. Translocation factor value of all compounds was higher in spinach than lettuce, and lettuce uptake was higher than spinach. In the constructed with mesocons experiment, plants were exposed to fast-packed solutions and wastewater. It was constituted of 4 batches. Each batch represented a specific solution treatment, 3 fast-packed solutions at different concentration, and a wastewater treatment plant effluent. In all batches, there were 3 replicates for species and 4 plants per tank. Macrophytes were exposed to contaminated PFAS solutions, Iris pseudochorus, Tifa latifolia, and Phragmites australis. This table shows the nominal concentration in nanogram per liter of FAS packaged solutions, hereafter called phyto and FAS concentration in wastewater. The experiment was performed 2 years ago from February to July. All tested waters were added of nutrients. At the end of the exposure period, we determined above ground and below ground biomass weight and FAS concentration, and FAS concentration in the expanded clay and the surface of the plastic tank. Considering the plant's response to the treatment, total dry weight of halophytes didn't decrease with the increasing FAS concentration. Dry biomass of iris and tifa significantly decreasing wastewater treatment plant effluent treatment, probably because of the high salt content. Bar graph shows FAS root concentration factor of each halophyte after the phyto 5000 and wastewater treatment period. We can clearly observe that FAS concentration in iris roots is generally greater than Phragmites and Tifa. Examining FAS above ground concentration profile, it decreased with increasing carbon chain length. Examining iris flower compartments treated by Phyto 2500 solution, we can appreciate the decrease of FAS concentration by disorder from stalk to flower, from flower to capsule, and from capsule to seeds. Furthermore, FAS concentration in all compartments decrease with increasing perfluorinated chain length. This bar chart shows the percentage of FAS removal efficiency of aquatic macrophytes and in particular the percentage ratio of FAS accumulated in leaves and roots of each halophyte. Removal efficiency grow with increasing FAS concentration, and Phragmites australis generally showed the greatest value, reaching 81%. Moreover, Phragmites accumulated in roots up to 88% of the total FAS removed. Using constructed wetland mesocosm experiment, vegetation didn't suffer the increasing FAS concentration in spiked solutions. Phragmites had the highest total removal efficiency from 27 to 81%, mostly in below-ground tissues. Tifa had the highest FAS removal in above-ground tissues. And talking about future perspectives, further investigations are needed. Give an example, investigation on further matrices like sludge and leachate, and investigation on long-term exposure to plant treatment in order to verify the FAS reallocation to roots. I am thankful to Professor Borin and Professor Dalferro for the precious assistance and the suggestions. Thank you very much to IMaSS Society Board for the opportunity. If you have any questions, comments, or suggestions for further investigations, you are welcome. You can contact us. Thank you very much.